Our first speaker today is Professor Lauren Ayton. Lauren is a professor of both optometry and ophthalmology. Lauren leads the Venture Register for Inherited Retinal Disease and is an investigator on several eye clinical trials. Today, Lauren will give us an introduction to inherited retinal disease and a broad overview of treatment pathways. Please welcome Lauren.
Great. Thank you so much for the kind introduction, Harrison. And a huge thank you to Anai and Ayton and all the organisers for the opportunity to come and speak to you today. We're incredibly privileged to be in this room together to share experiences and to give some updates on some of the research that's happening. And it's my pleasure to do a little bit of an introduction about what we mean when we talk about inherited retinal diseases. It can be a little bit confusing, so bear with me. We'll take you right back to the start. So when we're talking about the retina, it's the tissue at the back of the eye. It's like the film in the camera. And with inherited retinal diseases, there's problems in that retinal tissue due to changes in your genetic information. And inherited retinal diseases, or IRDs as we call them, are actually the most common cause of vision loss in people of working age in Australia. Interestingly, we don't know exactly how many people in Australia have these conditions, and we are actually trying to address that. So we've got research underway to try and find exactly how many people are affected, but we think it's around about 30,000 people. And what they do is they cause degeneration of vision over time. You'll sometimes hear researchers use the word monogenic, and it just means that it's caused by one single gene mutation. And that then opens up a lot of opportunities for treatments, which Tom will talk about a bit later. So some of you in the room might have age-related macular degeneration, and sometimes it can be a bit confusing because AMD also has genetic links. And so if there's a family history of AMD, people can also be at a higher risk of developing that. But the main difference is that AMD is what's called polygenic. So multiple genes are involved and there's environmental factors as well. And it's a more complex type of pathophysiology or causes of the vision loss. Macular degeneration is much more common, so it affects about 1 in 7 people over the age of 55, and about 17% of those people will lose vision. So there are differences between IRDs and AMD, but it can be a bit confusing to differentiate between them clinically. And some of you in the room may have had that experience where maybe you were told you had one and then were told you had the other. What has really changed that now is the ability for us to do genetic testing, and CC will talk to you a bit more about testing in a little while. So I've obviously got some slides up here. If you're not able to see them, I'm going to talk you through them. But basically, the retina at the back of the eye, someone has once said to me, it looks a little bit like a planet. So it's sort of a red colour and there's got a big circle in the middle, which is the nerve that takes the pictures from your eye back to your brain. And then the blood vessels radiate out from that. So in a normal retina, and the picture I've put up here is actually my own retina. So Tom, don't tell me there's a problem. I don't want to know. But it's basically a lovely pink kind of colour. What happens with inherited retinal diseases is we start to see changes in the retinal appearance. So we get black pigment clumps in the retina, the nerve itself gets a little bit paler, and we can see thinning of the blood vessels. The problem is that this is just one presentation of an inherited retinal disease. There are so many different ways that an IRD can present, and that makes it really complicated when it comes to diagnosis. So we now know of over 400 different related genes for an IRD. And to give you an indication, the first gene for an inherited retinal disease wasn't discovered until the 1980s. So we are rapidly learning more and more about the genes that cause these conditions. Oops, it's going to not want to play for me now. There we go. There are differences in the age of onset. So some people are born exhibiting the signs of an inherited retinal disease. Sometimes people don't show symptoms until they're even in their 70s. So there can be a really variable time that people start showing these changes. And the region of your vision that's affected can change as well. So sometimes it's the central vision that's affected. Sometimes it's the peripheral vision that's affected. Some people get patches of vision missing. There's all sorts of different experiences for people. And the other big difference is the progression. So there are inherited retinal diseases that are progressive and change over time, and there are some that are stationary and they don't change over the time. So you can see already the complexity when it comes to, first of all, diagnosing an inherited retinal disease, and then also giving the best advice as well, because there's so many different factors in here. As they're genetic, they are inherited in families. Sometimes in a family, there'll be very strong genetic links and people in the family will be quite aware of the condition. Sometimes they're a condition that is known as recessive, and so others in the family may not have shown that condition before. And the other thing with inherited retinal diseases is they can either be syndromic, and that means that other parts of the body are affected. And a really common example of this is Usher syndrome, where your hearing and your balance can also be affected as well as your vision. Or they may be non-syndromic, where it's just your vision being affected. And so some of the more common terms that you might have heard in the room is retinitis pigmentosa. So RP is one of the more common inherited retinal diseases. Sometimes we call that rod cone dystrophy as well. There is a flip of that, which is called cone rod dystrophy. And the easiest way to think about these two is the cone photoreceptor cells in your eye are responsible for your daylight vision and when it's quite bright, and your rod photoreceptors are more for nighttime vision. And there's more rods in the edge of your retina, there's more cones in the center of your retina. And so what it means is if the cones go first, the symptoms are different than if the rods go first. And the order of the name is the order it goes in. So cone rod means that you lose your central vision first. Rod cone means you lose your peripheral vision first, with some exceptions, of course, as always in medicine. Stargardt disease is another common name that you may have heard of. So this again is a central vision affecting inherited retinal disease. And again, when people go through a diagnostic journey, quite often you get a lot of these different names thrown at you. And hopefully the previous slide shows why that's the case, because there's so many complexities. And until we have a genetic diagnosis, we can't definitively say what it is. So I mentioned before Usher syndrome, and we do have Usher Kids Australia here today if you have more questions about that syndrome. There's another syndrome called Bardet-Biedl syndrome and Refsum syndrome as well. The reason it's really important for us to work out if it's a syndrome or if it's just affecting the eyes is sometimes there's different management if it's a syndrome. For example, Refsum syndrome actually can be helped by changing your diet. And so we need to know if that's what's happening so we can give people the best possible advice on what they should be doing for their health. So this morning we have a fantastic panel of speakers for you that are going to go over inherited retinal diseases in more detail. So first of all, we have Cece Britton-Jones, who very kindly has come in on maternity leave as well. So thank you, Cece. And she's going to talk to you about genetic testing and what it means to actually get that definitive answer from doing testing, and also why it is that we can't always give you the definitive answer. So she'll talk a little bit about what some of the barriers to being able to solve every single case at the moment. Parker is our genetic counsellor in our research team and also works at the Royal Melbourne Hospital, and some of you in the room have probably had the pleasure of speaking to Parker in the past. So Parker is going to talk us through what is genetic counselling, what's the process, what sort of questions should you be asking the genetic counsellor, and also ways that you can tap back into us, in both clinical and research settings if you have more questions as you go along. And then Tom's going to finish off this morning's session talking to us about the research pipeline that we have for inherited retinal diseases and also the clinical trials that are underway now as well. So Harrison in the introduction mentioned that Tom and I co-lead a project called the Venture Registry, which I know many of you in the room are involved in. And so this registry is a way for us to get people who are interested in research and trials on our database so that we can then let people know about the opportunities. There are similar registries around Australia. So there is a national registry, there's one in Sydney and one in Perth as well. And as I mentioned back at the start too, we also have some large-scale national projects happening where we're looking at mapping the different types of inherited retinal disease genes around the country as well. So if you're in the room and you haven't engaged in with research and you would like to, just talk to anyone that's here today. We are all very, very well connected and we can put you in the right place. So without further ado, I'm going to pass back over to Ayton so we can start chatting a little bit more about some of the scientific breakthroughs. But I'd just like to finish again by thanking you all so much for being here. As Ayton said, please do come and ask us as many questions as you like today or reach out to us later via email. We would love to be able to share what we're doing with you. Thank you so much.